Hi everyone, thank you for your time and thank you for your interest in my presentation on NLP and its role in drug discovery. So I'll be discussing about some of the NLP practices and case studies and success stories and limitations of NLP, one of the NLP technique called the generative AI and the transfer learning approaches. Thank you. Hope you find the presentation interesting. And this is my abstract for the today's talk. So majorly I'll be covering about the role of NLP in drug discovery and designing, and majorly I'll be discussing about how the NLP generative AI and transfer learning works in case of designing a new molecules and especially designing molecules with a desired property space and what are the limitations in terms of biological profile and success stories and so on. Thank you. Right. So let's start with a small introduction about the drug discovery workflow and some of the case studies how AI, ML, or NLP help us to improve the success rate. So drug discovery is basically a long and complex process that involves screening of thousands of compounds, or what we call it as a hits, to identify a potential drug-like candidate. And this process will take up to 10 years and cost billions of dollars. So let's take a closer look of the drug discovery workflow now here. So the basically the preclinical basic drug discovery is nothing but of designing a drug-like compounds. It has a multiple stages like hit and lead, identification and optimization. And during this process, we actually design thousands of compounds and synthesize them and screen them against the biological target of interest that is playing a key role in the therapeutic disorder. And we assume out of these thousands of compounds, some compounds will be uh, very much potent and lead-like compounds are ideal properties. So that will move to the preclinical stage for further testing. So this entire process will take around 3 to 6 years. Then, if out of 250 compounds, if one drug like molecule has ideal properties, that will move to the next stage, and we'll start with the clinical trials as phase one, phase two, phase three, with a different number of volunteers and with a different number of disease states and the normal healthy volunteers as well. So this will take around 6 to 7 years, and FDA approval will take around zero point 0.5 to 2 years. And average cost for designing a one molecule, for the entire drug discovery project will take around 10 years and cost $2.6 billion. So let's go into the closer look of the workflow, like what we discussed in the designing part. So, as you discussed about, it takes greater than 10 years and $2 billion. And especially when it comes to the molecule designing stage that has divided into target to hit or hit to lead or lead optimization, then followed by preclinical trials. And these are the, revenues and cycle time and cost per run that, contribute for that particular phase. So, and this actually indicates the success rate in the each phase. So you can see, you know, it's nothing but of how much success we have at the each stage, right? So now, if you see hit to clinical candidate, so it costs around 842 million dollars, and it costs around 5.5 years, and failure rate is 69 to 85 percentage. So it means that success rate is around 30 percentage. So to translating lab to market a compound. So after spending this much of $1 million, so you're not sure that drug-like candidate will move to the next phase, right? Now, we have a lot of approaches and lot of methods that is existing in designing a compound. And now let's see how AI, ML, or NLP works in case of drug discovery. So here, let's take a one of the let's take a look of the some of the success stories of AIML in drug discovery. So one such remarkable success story is the in silico medicine implementation of generative AI approach. It's called generative transferable reinforcement learning approach. So this approach allowed them to design a novel molecules for DDR1 kinases in relatively short amount of time. So in order to design the molecules and everything. So that they took around 21 days, and to synthesize and in vitro screening took around 46 days. And within the time span, so they were able to identify an nanomolar compound, not only the potency, they were also able to identify the compound as ideal PKPD parameters that they call it as a lead-like compound. So the traditional cycle of hit to lead that generally takes 2 to 3 years, and with the gentle approach or generative AI approach, they were able to design the molecule in less than 2 months. So now come back to the previous slide. As we discussed, the cost is involved around $800 million and 5.5 years and huge number of molecules to be designed at at the design phase. So now you are seeing using generative AI approach, they were able to identify a lead-like candidate within a span of 2 months and by synthesizing six compounds. So that is saving huge time, money, and reducing the failure ratio. Right. So that was one of the blockbuster success story, but it also has some concerns raised in the community called the molecule is so much closer to the one of the drug compound or one of the target dataset compounds. Yeah, but even though that's a novel approach that we can appreciate that. So when you break down the cycle, when you go into the design phase of this cycle, this 21 days, what happened? So they started with the understanding the existing kinase inhibitors or non-kinase set, X-ray target data set, and IP base, and they were able to identify a compounds which are known compound that used to train the model as what we call as a general, and so model training and followed by reward functions, and also added some more function called novelty. And by using general, they were able to design around 30,000 structures. And further, these 30,000 structures were processed by different filters like a pharmacophore analysis, docking, clustering, property analysis, different structural alerts, and followed by molecule commercial availability. So then they ended up having a 40 compounds, which are seems to be ideal, but based on the commercial availability and synthetic feasibility, they synthesized around six compounds, and they further expanded biology and synthesis. And out of which six compounds, the four compounds seems to be nanomolar compound, nanomolar potent, and PKPD parameters are you know are up to the mark. And another success story is from Excentia. Again, it's driven by the AI design compound, again as the A2A receptor antagonist. So it's a co-invented and developed by Excentia and Evotec. So Excentia designed the compound by leveraging its in-house AI-driven platform, and Evotec has provided biology and chemistry capabilities. And currently the compound is in clinical trials. And if you see the highlights of this approach, so they designed antagonist for the A2A receptor, which is very playing a key role in the triple negative breast cancer. And they set to identify the potent, highly selective compound against with A2A and with low CNS penetration. And the method they used is 2D evolution based on the fragment-based approaches. And this is one of the first AI approach they used for the A2A receptors. And they took around 9 months to generate a novel designs and to go to the DMT cycle. And they tested 163 compounds during the entire project. And they out of 163, one of the compound ended up into a clinical candidate. So you can see when you refer to the previous slides, so it took around you know uh, 5.5 years and followed by a large failure rate and followed by number of compound to be screened. And here you can see it's only 163 compounds here. And now coming to the another success story from Schrodinger. So they come up with a hit to clinical candidate within a short term, within a 10 months, and they were able to identify a discovery a novel potent inhibitor for MALT1. And again, it's a blood cancer molecule. And sorry. So for this MALT1 inhibitor, they screened around 8.2 billion compounds by using their in-house platform, and out of they shortlisted around 78 compounds, means during the DMT cycle, during the multiple iteration, they synthesized 78 compounds during the entire phase. And it took around 10 months for them to design the compound and make into clinical stage. And that's it. Out of 78 compounds, one compound uh, is moved into clinical stage now. And not only Schrodinger, Exscientia, or in silico medicine, there are many more success stories. Like you can see a lot of publications saying that they identified antibiotics, or they identified another cancer candidates, COVID molecule using AI approaches. Right. Now, before we move into the main topic, let's say have a basic understanding about how drug discovery works. So, drug discovery, especially, starts with the identification of a chemical space. So, chemical space is nothing but of is extremely vast, and it starts with the screening and filtering of this chemical space to identify the potential candidate that can be prioritized for synthesis and screening against the biological target. So, and coming to the this entire chemical space is almost entirely uncharted, and we don't have many data points to actually to consider and what to design next. And let's see how machine learning models or NLP or AI-based models, how helpful for us to identify right chemical space that suits our property profile and screen them in a faster and smarter way, right? So let's understand the terms here, biological terms. So biology target means the macromolecule of my interest, either it can be protein or RNA that involved in a disease pathway somehow. So the when we say compound or hit or lead or candidate, it's based on the stage we call it as either it's a hit compound or lead or clinical candidate. So it's nothing but of its bind and interact with the target. And affinity is nothing but of that actually, that term actually tells you the molecule is how much good in terms of sharing a complementarity with the target active site and binds and interact very well with the target. And toxicity makes sure the given molecule or designed molecule is not harmful to any living organism. Selectivity and make sure the compound is only binding to the desired target, desired biological target, not to anyone else, not to anyone, any other biological target to confirm off-target efforts. Not only this point, there is much more data points like solubility, COCO2, ADME, half-life, PKPD. So we need to search this chemical space that meets all these endpoints to identify best candidate, right? So basically what we are trying to do is we are identifying it from the chemical space. We are actually searching for using different techniques, whether we call it as a virtual screening or denovo designing or pharmacophore or structure-based virtual screening. And with a property space in your mind, like a stability, solubility, toxicity, off-target efforts, and that matches all this property threshold, and make sure we can consider these molecules for the next stage of synthesis and screening. And we assume one of the molecule make into drug that can either kill the virus or cancer. So based on the project objective is finally met the project objective, right? Okay, so here, sorry about that. Yeah. Now let's say, we want to design a molecule or search for the ideal candidate that matches our desired property space. So the one of the approach is direct way, the simulating by taking one of the compound and simulating them with the protein space and see whether that fits very well or not. That's called molecular dynamic simulation. But it takes days or months to screen one compound, to screen around huge number of nanoseconds. So when you want to do that for billions of compounds, it's not feasible. It might take years and computation space as well. So now next is nothing but of high throughput virtual screening. High throughput virtual screening is nothing but of either this can be structure based or analog based, is basically screening millions or billions of compounds uh, against a given pharmacophore model or structure based model. And within a day or weeks. So again, it's a timelines is depend on the computation power or space. So then finally processing those identified hits and cherry picking those hits, again time consuming process, and even procurement and synthesis and biological screening and will take months to years. And that's uh, about virtual screening. And now next comes to the denovo designing. Denovo designing is completely other way around. How? So it's nothing but before we design a molecule, we'll set a ideal parameters like what you want to achieve, like a drug-like parameters, target space, potency, safety, less toxic, or uh, no PANS filter, pains filter, and then we'll actually look for the molecule or design a molecule that matches this criteria. So one of the advantages and disadvantages of this method is we can actually do the selective design by using de novo approaches or either AI-based approaches, traditional de novo approaches. So major drawback of the de novo drug designing is synthetic feasibility. So yes, we can design the molecule, but we can't say whether they are synthetically available or feasible or whether it's a complex to synthesize. And whereas a virtual screening, we are actually taking the molecule that are available from the different commercial sources or vendors, and we can, as soon as we find the best candidate from the virtual screening and cherry picking them, we can actually go for the procurement or synthesis. Whereas in case of Dino, there is a major drawback. So we need to make sure like the given design molecule is able to synthesize fast. Right. Okay. Now let's come to the generative AI. So when it comes to the generative AI method, so we need to use the language-based models. So now for the language-based models, molecular representation and is very essential, and how computational powers read the molecule, the best way to encode and manipulate the chemical information by using different NLP libraries. So coming to the molecular representation, there are several types of representation like files, formula, 3D, graph, fragments, and the fragment-based approaches are electrostatic maps. Right. So one of the well-known you implemented technique is SMILES, taking the molecular information into the SMILE string. So SMILES are nothing but a simplified molecular input into a system that widely known as SMILES. So it's in human readable format that encodes the molecular structures using a series of rules, symbols, and strings and bond types, characters. So these smiles can be easily, can be searchable against the databases and to generate a molecules and to train the generative AI methods. Right. So using a different NLP technique, so these smiles can be readable and process the smiles for the generative AI experiments. So since the smiles format is encode the chemical structure that converts the smile strings into numerical format that can be input to the that can be given to the input as input to the generative AI models such as recurrent neural network or generative adversarial networks like GANs, and these models will learn a given chemical space or you know understand the given database as a latent space, and then understand the probability of distribution of the next characters, and then generate a novel molecules by given specified property ranges. So, and also that matches structurally similar to the given training set, and that can be used for the drug designing and other applications. Additionally, NLP can be used to analyze and classify the molecular structures and identifying the presence of specific functional groups or predicting molecular properties and toxicities, right? Basically, everything. So now comes to the key milestones in generative AI methods in drug discovery. So it's not only limited to the RNN based. There is a lot of methods has been proposed, autoencoders, GANs, flow, or conditional, and even for the fragment based approaches, scaffold based approaches, right? So even for the fragment linking approaches, sorry for the jumping. Right. So we'll discuss this one. and followed by generative AI implementations. So it's called, this plot indicates the relative frequency of, various AML models implementations and drug discovery. So you can see how it's growing year by year, right? So starts with a low one is RNN through GRU. So you can see how it is year by year. There are a lot of methods has been proposed are developing. So not only autoencoder scans or RNN LSTM is one of the widely used method for different NLP tasks in drug discovery. Right. Now let's move on to let's take an example of RNN and reinforcement method, how they are using RNN and uh, reinforcement techniques for the drug designing and how they design the molecules and what are the workflows. Right. So recurrent neural networks are types of a deep learning model that can be used for generative AI, especially text-based methods. So these RNNs are especially designed to process sequential data, as we have seen in the diagram, and process the sequential data such as sentences, sequence of characters, and generating a new sequences that's similar to the structure uh, given in the training set. RNNs are capable of long-term dependencies and data probabilities, and uh, that can generate a structure that has a coherent, similarity to the given training set and to the context. So one of the well-known example, you know, um, is smart, SMS. You can, when you are typing hi, so it actually predicts what's the next character. And similar to the smile, so when you start with a carbon as a first atom, so what is the next character? It gives a probability of what will be the tentative, probable to be next character, either nitrogen or oxygen or again the carbon itself. So in drug designing, RNA can be used to generate a novel molecule structures, and generally these RNAs are trained a database of smile strings, especially representing what we call it as a latent space. So generally, for most of the methods or most of the well-known methods in generative AI drug discovery methods is one of them is reinvent. So generally, we train these RNN models by using Campbell database as a latent space. So Campbell database is nothing but of a these are the Campbell has a curated database of compounds that are synthesized and tested against the various biological targets. So that means that the compound that are present in the Campbell means they are synthesizable and they are already biologically active. So we are training those compounds as a latent space for these models, generative models. And generative RNN can be, used for the different approaches to design a selective molecules or design random compounds or designing molecules with a certain property thresholds are structurally similar or structurally diverse or can be used for the linker development or fragment-based approaches by training the process by using maximum likelihood estimation, or you can also use the reinforcement techniques to define, train the RNN models to uh, maximize the reward signal with that represents the desired properties of the generated molecules. And reward signal can be based on various criteria such as predicted binding affinity or solubility or toxicity. This RNN models lends a generative molecule that maximizes the reward signals, which can lead to the identification of a better compound with the ideal drug-like characteristics or desired properties or desired biological affinity. So, in summary, RNNs are powerful deep learning techniques can be used that are implemented for the generative AI, generative chemistry. Right. So as I discussed, there are a lot of methods that has been proposed for for de novo designing uh, by using these approaches. So one of them is RNN. So one of them is reinvent workflows. So especially you can see the basic workflow is general prior is nothing but of this is a general database that has been trained on the Campbell. So the random sampling, once you do the random sampling and score them using desired properties or MPO score. Right. So this is a much more complex workflow. This is nothing but a general prior is nothing but a latent space or known biological chemical space, or we call it as a chembel. And small data set is nothing but of a target of interest. The molecules that are biologically tested are target of against a target of our interest. So that given by using a transfer learning approaches, this will learn the chemical space of a given biological target space, and then it will start designing molecules focusedly that matches that are similar to this given training set rather than random designing. Then do the sampling and followed by scoring. And this again approach is not a random, but it's a transfer by using a transfer learning. We're actually generating a molecule that are focused towards the biological target. And then, so it's similar to the previous workflow, but what we added is an agent. So this actually nothing but of the molecules that are generated by transfer learning against a small data set. Again, so reassessed by using a focused prior is nothing but of a target specific library and by a reward function called by agent, and also assessed by MPO score and any property threshold of interest. Then the reinforcement learning that gives an award that matches all these criteria and design a target specific or desired property threshold based molecules. Right. Then how do we do the benchmarking of these generated molecules? So there are a lot of benchmarking methods has been proposed, like Moses and even general. There is a different benchmarking methods has been proposed. Basically, this once the molecules are generated by these techniques, so we assess the molecules smiles whether they are valid or not, whether they are unique or how diverse to the compared to the training set, and how novel compared to the training set, how similar to the given training set, and whether they are synthetically feasible or not. For each, all the compounds that are generated by the RNN or RL-based approaches, and make sure we evaluate all these basic functionalities and make sure the compounds are valid and synthetically feasible and they are similar, not that much of similar, are diverse enough, are novel, and we'll take those compounds for the next stage of further evaluation and we'll consider them for synthesis and screening. And let's see some of the techniques that has been proposed that are some of the success stories. The first one of the success story from the University of Oxford. They've taken VGFR as a biological target, and then they have done a binding database search to identify the known chemical space or the known molecules that are reported against the target, and they used a character-based RNN, and they generated 10,000 compounds. They apply a lot of variable filters like solubility, synthesizability, similarity, and they selected 5 compounds to prove their proof of concept. And out of 5, the 2 compounds are active. And similar to the next one, ETH juries. So they used LSTM-based approaches to design a PPAR RXR agonist, and they designed 1,000 compounds with a COH as a starting tag, and they applied similarity or building blocks availability darkly score as a cutoff, and from this 1,000 they synthesized shortlisted 5 compounds that matches all these filters, and they screened for the biological activity. Out of 5, the 4 are active, starts with a nanomolar to micromolar range, and these are one of the top best compounds. And again, from in silico medicine, um, they've done one project in kinase panel, and they collected 30,000 compounds that are already kinase inhibitors from the Thomson database, and they used ATNC model, and they generated around 30,000 compounds as a smile strings, applied a lot of filters, and out of which they selected 50 compounds. Out of 50, 7 are active. So against a different kinase panel assays. And the next one again on the JAK1 or JAK2, the similar workflow, and the others has been selected. One compound, one is active and as a starting point. And similar to this DDR one we discussed, and they screened a huge library of kinases and known DDR1 active kinase molecules. And by using General, they're able to design 30,000 compounds and applied filters and shortlisted 6 compounds, out of which 4 are active and with a nanomolar potency. So this is all about how NLP and generative AI transfer learning approaches is able to help the drug discovery project. And and these are some of the success stories that is already published, are known, and now coming to the limitations. So generation in low data regime. So there are many success stories that we discussed. These models are trained on larger training sets. So we need to see, the new approaches are improved models that can, work in case of low data as well, and lack of unified evaluation protocol. Basically, once we generate a molecule, we use only whether the molecule is valid or unique or not. So we need to have a better evaluation protocols, and we need larger scale studies and benchmarking as well. And coming to the some are unrealistic for problem formulations, and some are ideal assumptions, and lack of interpretability. Once we generate a molecules, as I mentioned, but we need a lot of benchmarking studies, and we need a quantitatively bigger data sets or challenges that proves this model can work in either larger data sets or low data regime as well. So that's all about my presentation today. Thank you for your time, and I'm happy to hope you enjoyed my session, and I'm happy to answer any questions you have. Thank you so much, and looking forward for your questions.